Hello, thank you for joining us today to discuss accelerating COVID vaccine development with biosimulation. My name is Jian Cho. I am the Chief Strategy and Marketing Officer at Certara. Before we begin, a couple of housekeeping items. Next. You are in listen and view only mode for this session. There will be time for Q&A at the end. So along the way, please feel free to use the Q&A area to submit your questions and we will address them in the last 15 minutes. First, a little bit about Certara. Certara is the global leader in biosimulation with our gold standard software and tech-enabled services. Our biosimulation software platforms have been adopted by 11 regulatory agencies around the world. And we partner with top global pharmaceutical companies, as well as hundreds of biotechs and nonprofit organizations. In fact, 90% of new drugs approved by the US FDA since 2014 have been supported by Certara with our software and/or services. We are 850 strong with over 35 offices around the world, and we have deep scientific experts who are leaders in their fields. I am excited to introduce two of these leaders today. Thomas Kerbusch is Certara's Chief Growth Officer. He drives integrated growth opportunities with a special focus on executive client relations. During the last 25 years, Thomas has contributed to the development and approval of numerous drugs in a wide range of therapeutic indications, including cancer and HIV. Before joining Certara in 2014, he held leadership roles at Organon, Schering-Plough, Merck Sharp & Dohme, and Pfizer. Thomas received his undergraduate degree in biopharmaceutical sciences at Leiden University and Georgetown. He obtained his PhD in clinical pharmacology at the Netherlands Cancer Institute. He has co-authored or authored over 40 peer-reviewed publications. Next, Piet van der Graaf is our Senior Vice President of Quantitative Systems Pharmacology, leading all platform development. He brings over 20 years of experience working in the pharmaceutical industry with leadership roles at Sanofi and Pfizer before coming to Certara. He was founding editor-in-chief of CPT, Pharmacometrics and Systems Pharmacology, responsible for the successful launch of this open access journal, and he is currently editor-in-chief of Clinical Pharmacology and Therapeutics. He is also professor of Systems Pharmacology at Leiden University. Piet received his doctorate of pharmacy degree from the University of Groningen and a PhD in clinical medicine from King's College London. He is a fellow of the British Pharmacological Society and has authored or co-authored more than 150 papers and book chapters in quantitative and translational pharmacology. Piet also holds several patents in the field of target discovery. So today, Thomas and Piet will discuss challenges and considerations in vaccine development for COVID. What exactly is biosimulation and where is it heading? And then they're going to share two examples of biosimulation with model-based meta-analysis and quantitative systems pharmacology. So now I will turn it over to Thomas.
Thank you so much, Jen, and thank you very much for the opportunity to talk about this. So modeling and simulation, it's very common in innovative industries. A good example of that is building airplanes. No new airplane will take off before the whole airplane is first built virtually in the computer. Every nut and bolt, every experimental wing design, every engine part, and then in the computer, that construct is virtually tested. Landings in storms, engine failures, all before the actual building of the airplane and sending it in the sky. Building an airplane is a complex process, of course, but so too is developing drugs. The nuts and bolts in an airplane at least are known. The behavior of materials under stress is known. In contrast, the human body is much more complex and large parts are still poorly understood. Never mind understanding the best way to combat a new virus like SARS-CoV-2, which we'll talk about later today. I think you've seen probably similar graphical illustrations like this in different shapes and forms. Beyond a doubt, vaccines are the most effective medical interventions for saving human lives and preventing suffering. Hundreds of millions of lives have been saved through vaccines. They're all also very safe, the safest treatment modalities that we have. What used to be devastating diseases are now distant memories, except for maybe some vaccine cocktail jab you get as a baby. Despite this enormous success of the vaccines, they're actually notoriously hard to make. Ken Fraser, the CEO of Merck, recently made the point that we only managed to actually create 7 trillion novel vaccines in the last 25 years. Take Ebola, it took the medical community years to get a viable vaccine for Ebola. And for 30 years, they've tried to get vaccines for HIV and most forms of cancer and not succeed. And even when they do succeed in getting a vaccine, it doesn't always mean it's fully efficacious. Take the example of the flu vaccine. These vaccines are typically very safe and well tolerated. Each year, a new vaccine is created to address that year's strain of the virus. So it's a slightly different mutation every year. So you do have to create a new vaccine for it. Some years, the strain itself is more virulent, but we also have years where the vaccine that was developed is less effective in creating a good immune response and assuring the all-important herd immunity is achieved. And as a result, the effectiveness of the flu vaccine can actually differ substantially from year to year. 2018, for instance, was a typical year where we saw a lot more hospitalizations and deaths due to influenza. So the takeaway message here is, it's hard, and the medical community doesn't always get it right. So now let's talk about this simulated airplane, or actually how that relates to Certara and what we do. Certara simulates virtual patients in virtual clinical trials, the pressure test, the actual clinical trial before takeoff, before you conduct the trial. Think of Certara as the quants in drug development. We ourselves do not own or clinically test new drugs or vaccines. We help our 1,600 clients, the pharma and biotech companies across the world, with biosimulations to answer key questions that they face while developing their new drugs and vaccines. Biosimulation does not mean we make stuff up in the computer, but rather we build mathematical constructs of the available biological knowledge about disease, about the diseases, the human body, the effects that these drugs and vaccines have on the human body and on that disease. And then we use that construct in the computer to answer questions. Biosimulation has been a steady revolution in drug development, where over the course of the last 2 decades, we've seen it's become more and more prominent in key decision making in drug development. And Certara and its scientists are the key opinion leaders and the leaders in the field on this. As we start on COVID-19 vaccine development, most of these approaches are not as much tested for SARS-CoV-2, but biosimulation has been firmly established in drug development and also vaccine development programs to date. Vaccine development too needs biosimulation to get it right first time. Acceleration can be found in parallel activities, meaning you do a lot of the work at the same time, but also acknowledging the pharma companies don't always get it right and therefore need to simulate what success looks like. Trial and error is too costly, too slow. Failing first in the computer fast and cheap, and then we know what success looks like so that we can conduct the trial with certainty. It allows us to answer questions. How does the drug work? How do we cure or prevent the disease? What dose do we need to pick to cure or prevent the disease? And should it be different for different people? Biosimulation also can select the best vaccine candidates to prepare for the all-important manufacturing scale-up process. We'll talk about that later. It will also allow you to pick the best vaccines from the many candidates entering testing. Pick the winners. More on that later too. So how do we do that? Certara builds virtual patient groups, for instance, for different age groups or racial backgrounds or specific patient populations. We build these virtual patient platforms in collaboration with our pharma clients. An example is our SimSip biosimulation platform that has been developed in a consortium with 35 leading pharma companies. These platforms have broad acceptance. The regulatory agencies over the years have confirmed the validity of these virtual patient platforms and use them in reviewing new drug applications. It allows them to ask what-if questions. Despite the extensive clinical testing, not all scenarios can be studied during clinical development, and some data may be underrepresented. For instance, what about a 100-pound, 80-year-old woman of Japanese descent? Will she respond differently to a drug compared to, say, a 40-year-old white male of 200 pounds? And what about people with organ deficiencies like kidney disease? Will they clear the drug sufficiently? And what about if we combine drugs in the patients? Would we see drug-drug interactions? All of this can be answered through virtual patients. As we understand the underlying biology that makes all these people different, we can extrapolate and predict where problems may occur. In situations like that, the FDA may ask for additional clinical data or when the models indicate that there are no differences to be expected, no additional trials are required, and further R&D costs is avoided, and it assures that patients that need these drugs will get access to these drugs sooner. To date, Certara is helping their clients, companies in over 20 programs for COVID. These are global large pharma and biotech companies. We help them on programs for active treatment with repurposed drugs, and now also with new drug candidates specifically designed for the SARS-CoV-2 vaccine, SARS-CoV-2 virus. Much of this work is done through the COVID-19 Therapeutics Accelerator Initiative, led by the Bill and Melinda Gates Foundation. We're basically their biosimulation department. Certara is also partnering with DMTC Limited, the company leading Australia's medical countermeasures program, to provide independent technical evaluations, product development and risk management advice to the Queensland government with regard to a potential COVID-19 vaccine candidate under development in Queensland. Ultimately, we see the active treatment of a viral infection will need a combination therapy approach, not dissimilar to HIV and HCV, or like the combination therapies now applied in cancer, including immuno-oncology. We're currently thinking how we can accelerate that development. For HIV, it took decades to get an effective combination therapy, and we've learned from that. Biosimulation will allow for less trial and error testing in the clinic and fast forward the development of what we would call a polypill solution. Anybody knows what R&D is, but most people actually think mostly about the research part, discovering novel mechanisms, vaccine platforms that deploy different means of triggering a protective immunological response and immune memory. This is all cutting edge research and very exciting. But there's also the testing part, the development part. First in the lab, in petri dishes and animals, then we test in humans. And this development process is centered around a series of key questions. We want to know if it's safe. Development programs logically focus on safety foremost. If we're going to expose billions of what are essentially healthy people, we better make sure that it's safe always. Because after phase 1, where we look at safety and tolerability, and phase 2, where we look at safety, tolerability, and immunological response, we typically enter phase 3 in very large trials where we test prophylactic efficacy and we look at safety again. And at that point, we also make choices around our dose. Do we have the right dosing regimen? Is it optimized for a lasting immune response? Do we need to revaccinate, and how often? Do elderly and the frail need a different dose? We know that their immune response is likely less strong, and therefore they may need a different dosing strength. How about children, pregnant women? Do frontline healthcare workers require the same dose? All critical questions. And biosimulation is essential in all steps. Accelerating vaccines for COVID-19 should not be about creating a safe vaccine only, but also, is it safe and efficacious? Getting it right first time round, not the second, third, or fourth shot on goal. Production cost and medical urgency is simply too high for that, to allow for multiple shots on goal. As you know, COVID-19 is being developed under unprecedented timelines. This is achieved by running most activities in parallel, condensing it down to approximately 18 months, if a vaccine candidate indeed is successful in every testing step. That is why it's so important that we have multiple vaccines being developed simultaneously at the moment. I would like to call out a critical piece though, the manufacturing. Setting up production lines, allowing for this magnitude of production costs billions. This is also done at risk. Think about that. Often the academics take the limelight around the new science, but the pharma companies really do stick their necks out here by investing billions in basically setting up production for vaccines that have not been tested yet in the clinic. The problem is that if we fail in phase 3, and it happens, if we fail in phase 3, we can't just simply switch the manufacturing plant over to another vaccine candidate. Manufacturing needs to be reproducible, precise every single time. This means that production processes are validated and tested endlessly, but specifically for that vaccine and that vaccine dose. That is really where the cost sits in manufacturing. The consequence is that early on in the clinical development of a new vaccine, the dose is locked in, where in a normal paradigm you have time to explore the dose selection. Here now, that time frame is very much condensed. As a consequence, we may not always have the most optimal dose for both safety and efficacy. And this brings us back where we come in, Certara's biosimulation. Biosimulations does allow you to ask these questions before you trigger those investment steps. Biosimulations allows you to get it right first time round. Good news is that we currently have around 170 vaccine candidates in development, and this number changes every day. So that's very exciting. The majority are still in preclinical testing, but there's about 37 of them in clinical testing, and 6 now have ended in large-scale trials. But are they the right vaccines for the right people? We know that the immune system is different for instance, the elderly and the frail, which is the exact most vulnerable group for COVID-19. We also know that children are not miniature adults, especially neonates and infants. Their organs are developing and therefore often have reduced organ function and therefore need different doses. And how do we compare vaccines from this large mass of clinical trial data? These trials are not carbon copies of each other and will differ in how the data is gathered, which types of subjects are included, for how long are subjects observed in the trial. This may all differ from trial to trial. And how do we compare and this is specifically where our biosimulations can help gather that information. So let's talk about that. There's a biosimulation type called a model-based meta-analysis, MBMA for short. It's a form of statistical network analysis that uses large databases across many compounds in clinical trials. Certara has been pioneering this for the last 15 years and is the market leader in this highly impactful form of biosimulation. Certara gathers all available data on clinical trials from scientific publications. We have developed curated and highly reproducible database creation processes enabled by AI-based technologies. Certara now provides and maintains over 45 clinical outcomes databases for their clients. Some of these databases contain over 1,200 randomized controlled clinical trials for an indication. We then use that data to run biosimulations where we describe the treatment outcome, efficacy, or safety across compounds or vaccines within a specific therapeutic indication. As trials typically compare against placebo, we can compare indirectly between, for instance, vaccines that were never really studied head-to-head in the same trial. We then estimate the underlying effects of, for instance, background therapies, co-medication, disease severity, patient characteristics, the length of the trial that people are studied in, and so on. All those differences that we may observe between those trials, we try to estimate what the impact of those factors are. Some trials may have sicker patients in there, and if people are sicker, it may be easier to demonstrate an effect, for instance. So when we compare the vaccines, you want to make sure that we compare apples to apples, and we correct for that. And it allows for a head-to-head comparison of virtual patients in a virtual trial. It allows you to run this virtual head-to-head comparison before you run the actual trial. And we've seen examples over the years where trials worth and costing hundreds of millions of dollars were basically doomed to fail if you would execute them. We would, through biosimulation, indicate that would be a flip of the coin and that you would be better than the standard of care, and you may very likely actually perform worse than an emerging competitor from another company. You really want to run that trial. It's a very costly way of demonstrating that you're less good and your competitor is better. We've now also built a database for our COVID trials. This was sponsored by the Bill and Melinda Gates Foundation, and currently it's focused on the active treatments because that's where most of the available data now sits. But very soon, more vaccine data will start to emerge from these clinical trials. We'll update the database with that. With so many emerging candidates, soon we'll have dozens of trials. How do we then pick the best? How do we correct for those differences in trial designs, dosing regimens, adjuvant use, special populations? Say in one population, in one trial, we have more young people than in another trial with another vaccine where there's more elderly people, which we do expect to have less of an immune response. MBMA can estimate the differences between those different trials and allow for an apples-to-apples comparison. Regulators and payers will be very interested in those comparisons to underpin the choices they make in approving and paying for vaccines. And will actually also help the drug development companies by showing them how initial immune responses in healthy young populations ultimately translate to this protection of the more vulnerable populations in phase 3. Say there's a front-runner vaccine that has gone through phase 2, and from phase 2 we have immunological response data. We also have phase 3 data from that front-runner vaccine, phase 3 data on the ability to protect against the infection from SARS-CoV-2. And now a second vaccine enters development, and we get phase 2 data, and we see the immunological response, and say that immunological response is 10% better than the front-runner. We can translate through model-based meta-analysis what sort of improvement on preventing the infection rate in phase 3 we can see. That means, does it really have sufficient clinical significance? Is it worth preparing manufacturing for at that point? Critical investment questions for pharma companies. So where model-based meta-analysis is a biosimulation technique where we make inference from emerging clinical data, I now want to hand it over to my colleague Piet van der Graaf, who will tell us about quantitative systems pharmacology, a type of biosimulation that predicts if a drug will work before we even have clinical data.
Thank you, Thomas. The most recent development in biosimulation is called Quantitative Systems Pharmacology, or QSP. As the name indicates, this approach aims to develop mathematical models of the whole system or body. These models represent human biology and disease in great detail, and because biology is complex, the models are complex and often very large, with thousands of mathematical equations. 4 years ago at Certara, we embarked on the development of such a QSP platform for the human immune system in a consortium with 7 major pharmaceutical companies. The initial focus was not on vaccines, but on the immune response that is often seen following administration of therapeutic protein medicines. This so-called immunogenic response is highly undesirable since it significantly reduces the efficacy, often so badly that a patient has to switch to another medicine. It is highly variable, which not only differs between medicines, but also between different patients, age, gender, and ethnic groups. In partnership with the 7 pharma companies, we have now developed for the first time a model which has been validated with more than 20 clinical case studies that can not only predict this phenomenon, but perhaps more importantly, help to reduce its impacts through, for example, changing the dosing schedule. More recently, we realized that the same model can also be used for vaccine development. Our model predicts the human immunological response to the administration of an antigen. In the case where the antigen is a therapeutic protein, the immune response is undesirable and needs to be minimized as much as possible. In the case of vaccines, the immune response to the antigen is the intended effect and needs to be enhanced as much as possible. So when the COVID crisis hit the world, we put a lot of effort into repurposing our immunology QSP platform towards vaccines and validated its predictive abilities using existing vaccines, such as the ones for hepatitis B. Encouraged by the early results, we then developed a prototype model for COVID-19. And I'm very excited that we have just started a collaboration with a major pharmaceutical company to fast-track the development of their COVID-19 vaccine using our QSP platform model. In the next slide, I will explain how we can address key questions such as what is the optimal dose in different population groups using so-called virtual patients before testing the vaccine in actual patients. The idea is that we can use our model to create virtual patients on whom we can test a novel vaccine on the computer before actual clinical testing. You can think of a virtual patient as an in silico clone of an actual patient on the computer. Differences between actual patients can be represented in the model, such as genetics, gender, ethnicity, age, disease, and medication. For example, genetic variation called alleles can play an important role in the response to a vaccine, and our computer model can predict such differences, for example, between a Northern European and an Asian population. With our model, we can generate not just one, but hundreds or thousands of different virtual patients, which could, for example, reflect the expected demographic composition of a phase 3 clinical trial. With this group of virtual patients, we can now run virtual trials, testing, for example, a range of different doses. It would take months, if not years, to do this in actual clinical trials. On the computer, it takes hours. Virtual patients and virtual trials can be used at all stages of vaccine development. Before the first administration to humans, we can first determine with the model what the optimal structure of the vaccine is and what the effect is of co-administration of so-called adjuvants, which are molecules that give an extra boost to the immune response and can therefore enhance the effectiveness of the vaccine. Next, we can use the QSP model to determine the optimal dose and number of shots that should be administered in a phase 1 trial in healthy volunteers and the time between them. Next, when the first actual clinical data in healthy volunteers are obtained, the model can be refined and then optimized, and then virtual patients can be created for specific patient groups, such as elderly, children, pregnant women, and different ethnic groups. For example, as was recently pointed out in an editorial in the New England Journal of Medicine by Dr. Penny Heaton, the CEO of the Bill and Melinda Gates Medical Research Institute, elderly adults may require a higher dose, as observed with the influenza flu vaccine. This is a good example of the kind of critical questions that can be answered with our QSP model. So let's see what our model predicts for a COVID-19 vaccine for young versus older virtual patients. What you see here are simulations with our QSP model of the response to a hypothetical COVID vaccine in 2 virtual patient groups, the young on the left and the elderly on the right. The model predicts a strong immune response in the younger group, where the first dose is already effective in 80% of the patients, with an additional 5% improvement predicted after the second shot. In contrast, the effect of the first dose in the elderly is significantly less, reaching only 60% effect, whereas the second dose takes a response to almost 80%. This illustrates that it is unlikely there is a one-size-fits-all approach that works for everyone, and our model can be used to ensure every individual patient gets the best possible treatment. And I would now like to hand it back to Thomas for the concluding remarks.
Thank you, Piet. Yeah, so as we work with so many different companies, we have the privilege of being at the front seat here, seeing the science in COVID unfold. Very exciting. It also means that we can provide our solutions, our biosimulations to those programs and how vaccines are being developed. Of course, we have a long track record in using biosimulations where virtual patients allow us to select the right dose and dosing regimen. We can now help inform the same thing for the vaccines for COVID-19. Design the most optimal trial, get the answers sooner. Speed is of the essence. Speed is gotten by getting it right first time around. First fail on the computer so you don't have to fail in your actual clinical trials. Yes, vaccines always need to be safe, but they also need to work well enough, not just a little. The urgency of the medical need is too high to not answer key questions such as, do we have the right dose and regimen to get optimal level of duration of prophylactic response? And if not, do we need to revaccinate, use initial booster regimens, adjuvants, and do all subpopulations such as the elderly and people with underlying diseases, comorbidities require the same regimen? Dosing matters. Dosing the right dose at the right time for the right patient. And vaccines too will benefit tremendously from biosimulations. Specifically, model-based meta-analysis is a powerful tool to translate immunological responses to prophylactic protection and conduct comparative effectiveness assessments in our search for the best vaccine for global scale-up and rollout. Pick the winners. Quantitative systems pharmacology, as Piet demonstrated, originally established for unwanted immunogenicity of biologics, can now be repurposed to predict wanted immunogenicity responses of novel vaccines. We also strongly believe we have a huge opportunity, not just for this pandemic, but to be better prepared for when, not if, when the next pandemic hits. It may sound weird, but we are actually quite enthusiastic in the face of this pandemic. We're excited because we know that we can provide a difference here. We know we can provide smarter solutions and making sure that vaccines are accelerated to patients faster, safe, and efficacious. Getting it right first time around. And with that, I would like to wrap it up and open it up for Q&A. I see that there is a question has come in here. Okay, so I'll read out the question. I'd be interested in hearing what the utility biosimulation may have for reducing the complexity of inclusion and exclusion criteria in evaluating human subjects. It's a very good question. So how can biosimulation help in setting inclusion and exclusion criteria in clinical trials? As we look at the impact of what inclusion and exclusion criteria does, meaning do you limit your trial to less different types of people, or do you expand it to more types of people? That will have an impact on the outcome of the trial. It will have an impact on what you're trying to demonstrate on the efficacy, potentially also on the safety. And typical examples are, for instance, people that smoke. Do you include or not include them in certain trials where we know they may have an upregulated metabolism, and that may have an impact on drug levels, that may have an impact on efficacy. So yes, biosimulation very much is being used for that as well. Let's see another question here, and I'll read it out as well. Unless Piet, did you have anything to build on that? Anything else to add to that?
No, I think you covered it really well, and I think the concept that I talked about regarding virtual patients speaks to directly to this question. So we can obviously create virtual patients with very different backgrounds that we can use to help quantify and define inclusion exclusion criteria in actual trials. Yeah.
Yeah. Another question here, and I'll read it out. We see phase 3 clinical trials enrolling thousands of healthy individuals. Can biosimulation help reduce these numbers? I think yes, and let me answer that why I think that is the case. First of all, we're interested in these very large trials to demonstrate if something is safe. We also do want to know if it is efficacious or not. And so where biosimulation can indicate which of the different patient types, healthy volunteer types in the situation of COVID, of course, would have more or less of a response, you then want to make sure that you have enough of those people in your trial as well to be sure to test that. So if you end up in a trial where there may be an underrepresentation of certain subgroups that doesn't allow you to come up with conclusive answers at the end, that would not be a good trial. And so biosimulation can really inform on that to begin with. Now, I must also say on the counter side of that, for a large phase 3 trial, safety needs to be gathered. And sometimes with vaccines, and again, I'm going to repeat, they are the most safe treatment modality developed, but at the same time, rare things may happen, and for that you need to study large populations. And that's one of the reasons why you see clinical trials, phase 3 trials now with over 30,000 subjects. Piet, also handing it back to you. Any comments on this question?
No, it's a very good question, and I think you already covered it. But I think also maybe going back to your earlier point about that we need to get this right the first time. So it's not just a question about number of individuals in a single trial. It's also kind of how often would you need to repeat that trial if you don't get it right. And I think as hopefully we have shown, with our biosimulation, we can really help to ensure that such very large trials are designed in the best possible manner to get it right the first time. For example, making sure that we test the right dose in the right patient group.
Yeah, and I think you touch upon a point that we did mention during the presentation part as well. As we've worked for decades in drug development, we know often drug development programs fail to demonstrate efficacy, fail to demonstrate that they're safe, and that means that typically it is an iterative process, and at times you then have to go back to the next compound in development in there. And so knowing where we fail in those programs actually is also something where additional learning is obtained, which is then captured into the models, and the models then iteratively get updated. As I said, the models are a capturing of the knowledge that is available at that time. And we see with COVID, it's very much an expanding field. The science is emerging every day. And so the whole discussion around the role of the B cells and the T cells and the memory component, those are, I would say, state-of-the-art biological discussions that we then build into our models, that we then make sure that we can use in there as well. Let's see. I see another question here. So there's a question around the use of artificial intelligence, and do we use artificial intelligence? And yes, we do, and we use that specifically in the data gathering and data curation part. Some of the modeling processes are also steered through algorithms, not so much self-learning algorithms, but they do make assessments and they do update themselves. Artificial intelligence is actually a very broad subject term, and underneath that there's many different other terms. When it comes down to data searching, machine learning is probably the thing that we use the most. And so gathering information from these clinical trials actually means we go out and we get all the publications, all the randomized controlled trials, and then we use AI to read through them and collect the information and pull that information contextually together. There is a human curation part around that as well, and I think that's quite important to point out. Big data is another term that's often used, and I want to dispel that a little bit. Big data typically is you gather as much data as possible, and then you troll for some form of knowledge in there. Versus we build biosimulations to build models of what we know is the biology, what we read up is how drugs work and how the human body works, to then feed that with as much data as possible. But we go out in a very structured way how we want to get that data, how we collect that data. And that's a very structured process, means it's balanced, it's reproducible, and it is enabled by AI. It makes it a lot faster, it makes it more comprehensive as well. Piet, sorry, also looking at you, if there's anything else?
Yeah, yeah, so maybe another example of how we use that is in our QSP model and the way we feed in the properties of the antigen. So in the case of, say, COVID, people have been exploring the famous spike protein, and so what we do is we feed the structure of the spike protein or any other part of the virus into our model, and we use machine learning to infer from the structure, so the actual protein structure, how the human immune system is going to respond to that. So we use a whole suite of very advanced machine learning informatics tools to do that, to bridge between the structure of the protein and the response of the human system, mainly because there obviously is often very, very little actual data on that. So we have to use other approaches such as AI and machine learning for that.
Another question here, can biosimulation be used to help reduce the risk for manufacturing? And I would say absolutely. This is the part where the quantitative systems pharmacology models from Piet allow you to know which doses are the best. We observe under these accelerated timelines that those exploration, meaning testing multiple doses, figuring out what's the most optimal dose, is done, I would say, faster than normal. And as a result, not always the most optimal dose is carried forward from the efficacy perspective. It will be safe going into phase 3, but it may not be optimized on efficacy. And as I said in that schedule slide, where we basically have the manufacturing start in parallel with the clinical development, that time frame is very much squeezed. The ability to do dose exploration in the early trials, seeing what the most optimal dose is for both efficacy and safety, is very curtailed. And so the models from Piet really help in informing that, in optimizing that dose, and then inform the manufacturing process. And again, the manufacturing process, it's of course, they need to build a factory, but it's the validation process, it's the testing of the equipment before the actual productions go out, the stability of the product. That all takes a very long time. And for that, they need to know the dose strength. So I absolutely do think you can de-risk it. We do totally understand it needs to be accelerated and be in parallel. And that's the reason why you then go simulate it in the computer. Biosimulation does allow you to test it relatively rapidly. And so where a clinical trial may run for weeks for phase 1 trials and many, many months for phase 3 trials, computer simulations of thousands of virtual patients can be done in hours. And so that's a lot faster. Piet, again, handing it over to you as well.
Yeah. I mean, although our specifically our QSP model doesn't currently include, actual manufacturing, but I think the question, can it help reduce the risk for manufacturing? As Thomas said, well, absolutely. I think the models really help you to invest in the right manufacturing for the right vaccine. And also, for example, it would make quite a significant difference in terms of, the capacity required if we need 1 shot, 2 shots, or maybe even 3 shots of a specific vaccine. So I think, indirectly, the modeling does certainly help, optimizing investments in the manufacturing to make sure it invests in the right vaccine and at the right scale.
Okay, so I think these are the questions that we have received so far. Unless there's any other questions, please do forward them now, and I'll wait a few seconds to see if anything else comes in. If not, we can wrap up. Okay, if no further questions, I'll hand it back up to Jun for the last wrap up around this presentation. Thank you.
Thank you very much for coming. We really appreciate your engagement and questions, and we'll follow up individually to address any other questions you might have. So thank you. And thanks, Thomas and Piet.
It was a pleasure.